Now the stomach is going to contain all four of the tunics, plus it's going to have that extra muscular layer that we don't see anywhere else. Also, we're going to see some modification in the mucosa in order to deal with a really, really hostile environment that we have in the stomach. The muscularis externa is going to have that circular and the longitudinal layers that we see all throughout the alimentary canal, but it's also going to contain that third layer, which is the inner oblique layer that allows the stomach to basically wring and churn and mix our food with the digestive enzymes that the bolus is going to come in contact with once it reaches the stomach. So we could say then that our stomach is a site of chemical digestion as well as mechanical digestion. Now the mucosal layer is going to be modified. It's going to consist of the simple columnar epithelium, it's going to secrete this interesting two-layer coat. So it's an alkaline mucus, and the reason why we need an alkaline mucus lining our epithelial tissue in the stomach is because we've got cells in the stomach that are going to produce hydrochloric acid. So this interesting mucosal layer is going to protect the cells of the stomach, and it's also going to trap bicarbonate-rich fluid in between those two layers. So you're going to have a mucus layer that's immediately adjacent to the simple columnar epithelial cells of the epithelial lining of the stomach. Then you're going to have a bicarbonate-rich fluid layer, and then on top of that you're going to have another mucus layer. And we have to have that in place in order to protect the stomach from the enzymes that it's making in order to begin the digestion of food. Now, dotting the stomach wall are going to be these pits. They're called gastric pits. And down in the pits are going to be the cells or the glands that are making the gastric juices of the stomach. So there are 4 gland cells we'd like for you to know something about that are going to be in these gastric pits. And we're going to have mucous neck cells, parietal cells, chief cells, and enteroendocrine cells. Now, a couple of these are pretty easy for us to figure out what they're doing. So a mucus cell is going to be making mucus, and I'll tell you when we get to the picture why it's called the neck cell. And the endocrine cells are going to be making hormones. We just don't know exactly for sure which ones. Chief cells and parietal cells, no, we might not know so much about them, but we will in just a minute. So let's take a peek at this picture before we get into what these different cells are doing. So we have our mucous neck cells right here. Now the reason why these are called mucus cells is because of what they make, but they're called neck cells because they appear here in this narrowing of the gastric pit. And we know that anywhere in anatomy where we have a narrowing of a structure, it's called the neck. So that's why they're called mucous neck cells. The parietal cells are found off to the sides here, and which is interesting because the chief cells are too. And I know that the word chief is associated with the word gastric. I don't really know what the connection is. Parietal means off to the side. Chief is just associated with gastric. And then down here in the bottom, we're going to have where are our endocrine cells? Endocrine, right here, the green ones. The enteroendocrine cells right down there. So this kind of gives you an idea of where we're going to find the different cells that we're about to take a look at and see the different things that they're making for us. So the mucous neck cells are going to secrete mucus, and we say we don't really know what its function is, or why it's acidic. If I had to speculate, I would say the acidity is helping to break down the proteins, because that's what hydrochloric acid is targeting, and its mucus probably provides some sort of protection. If I had to speculate, that's where I would go with that. Although it says here we don't really know. The parietal cells, they're going to secrete a couple of things. This is where we're making the hydrochloric acid, and we're also making another really important substance known as intrinsic factor. Now, intrinsic factor is something that you definitely want to know about. Intrinsic factor is a glycoprotein that is necessary for the absorption of vitamin B12. We have to have B12. Why do we need vitamin B12? We need B12 in order to make red blood cells. So if we don't have intrinsic factor in our stomach, then we can't absorb vitamin B12, and that leads to a condition known as pernicious anemia. And by the way, where do we get vitamin B12? Largely from meat. Not entirely, but largely. Now, chief cells are going to be found a little bit deeper in the gastric pits, and they're going to secrete an enzyme known as pepsinogen. Now, you might have noticed that when Mother Nature releases a molecule or a chemical in the body that is almost ready to go but not in its active form, we add the ending ogen to it. So this is what we've got going on here. We've got pepsinogen being released, and then it's going to have to make its way out of that gastric pit and then get into the stomach. Now, why wouldn't we release an active form of that? Because we're right here in the stomach. I'll tell you why. Because this is some nasty stuff. And if it were to be released in its active form, it would digest the chief cell and anything else it hit along its way, trying to make its way out of the gastric pit. So we release it in an almost ready to go form. And the thing that activates it is the hydrochloric acid in the stomach that was made by the other cell, the parietal cell. Okay, so hydrochloric acid is going to activate pepsinogen to become active once it hits the stomach. Another thing that's going to be released here by the chief cells are a variety of lipases, enzymes that are going to break down fat. So we start that process here in the stomach as well. That is the breakdown of fats. Then we have the enteroendocrine cells, which are going to be releasing chemical messengers that are going to communicate with the stomach and maybe even some other cells in the intestinal tract. So gastrin is one of those. It's going to stimulate the parietal and chief cells to release their secretions. So down at the very bottom of the pits where we got these enteroendocrine cells, and they're going to be basically the ones that are saying, all right, go, release your things. We're ready. Gastrin is also going to stimulate the gallbladder and the pancreas to release their secretions. So gastrin will cause the gallbladder to release bile, and it'll also cause the pancreas to release its concoction of digestive enzymes. It'll also trigger the digestive tract to start moving, because basically once we're releasing enzymes in the stomach, it's a signal, that's the green flag that says the race is on. Food's coming. Everybody get ready. All right. And it can even cause the ileocecal valve to relax, possibly as an opportunity to allow food, any residue that's in the lower part of the intestine, to make its way out in preparation of what's coming in. Serotonin is another molecule that gets released by the enteroendocrine cells of the gastric pit, and it's going to work locally on the stomach muscle. Basically, this serotonin is going to trigger the three layers of the muscle of the stomach to contract and start doing that mechanical digestion. So it gets triggered for release when there's something in the stomach. Histamine is also going to be locally active. Let me just point this out here. You'll notice that I have the word paracrine here. Paracrine is a chemical messenger that gets released and is intended to act on local tissues. So para means near or adjacent to or next to. So the enteroendocrine cells are going to be releasing these molecules so that it will work on the tissues of the stomach. It's not intended to enter in the bloodstream and go elsewhere. It's not intended to be part of the concoction that makes its way into the stomach and may signal some other cells. It's not intended for that. Serotonin and histamines are intended to be local molecules having local effect. So stimulating muscle contractions and further stimulating parietal cells to release hydrochloric acid along with gastrin. Somatostatin is kind of like a braking mechanism, and it can, if it gets released, then it's going to restrict secretion and actually reduce absorption. in the stomach. So remember, wherever Mother Nature gives you a gas pedal, she's also going to give you a brake. And in the stomach, somatostatin is the braking mechanism. In the stomach, the three layers of muscle, that outer longitudinal layer, the inner circular layer, and then the inner oblique layer, that other one, all of those are allowing for mechanical digestion. So it's allowing the bolus to basically be converted into chyme as it mixes with the secretions from the chief and the parietal cells and the mucous neck cells. Chemical digestion is going to occur when the secretions from the gastric pits starts to work on the bolus and turning it into chyme and breaking it down at the molecular level. That's chemical digestion. Now, we've already talked about that special mucosal barrier that we see in the stomach, that double layer bicarbonate-rich mucus barrier that the stomach has, but let's go ahead and talk about it anyway. So we know that we have that. So we have one layer of mucus, and we have this bicarbonate-rich fluid, which we know the bicarbonate molecule is a molecule that the body uses as a buffer because it can reduce acidity. So that's the molecule that we see in our blood as part of our blood buffer. And it's used here as well to buffer the pH of the stomach. And then we have a second layer on top of that bicarbonate-rich fluid. So we create this really thick layer that is all designed to protect the epithelial cells. Now, the other thing that the stomach has in place are tight junctions between epithelial cells. And if you don't remember what those are contact points between the cells that cause the cells to be strongly adhered together so that nothing can sneak past the cells, because we really don't want this stomach acid and the enzymes in the stomach to get in between the cells and get down into the deeper tissues, because it'll digest those tissues. Now, as if those things weren't enough, this extra heavy-duty mucus layer plus the tight junctions of the epithelial cells in the stomach are replaced every 3 to 6 days, because Mother Nature knew that this was going to be a rough environment, that the chances of damage would lead to, that there's chance of damage, and that any damage would lead to far greater damage. So she just made these rapidly turning over cells in the stomach. So epithelial cells have a tendency to do that anyway, but in the stomach it's super fast. All designed to protect the deeper tissues of the body from the digestive juices of the stomach. Now, the small intestine has some interesting features in it that are designed to make it a super sponge, a super absorbent sponge. And these modifications include circular folds, villi, and microvilli. So let's take a look at these. So the circular folds are these permanent folds in the wall of the intestinal tract. They're about a ½ inch deep, about a centimeter deep. And the way that they go around the lumen, and the way that they go around the lumen basically forces the chyme to spiral through the intestinal tract. So as the chyme goes through the intestinal tract, it basically has to go up and down over these circular folds. And the circular folds are going to add to this surface area of the intestinal tract, which is going to increase the surface area for absorption. It's also going to slow the chyme down, so it doesn't fly through the intestinal tract, but it lingers and allows time for the enzymes to work and for the absorption to happen. Now, the mucosa is also going to have these finger-like projections called villi, and they're pretty small. They're about a millimeter high, so they're not like, super duper big like cilia, something like that. But they're these finger-like projections that are in the mucosa, and they're anchored in place by actin, which is a type of a protein, which we see in muscle, since we're put it out there. But it holds these villi projections in place. And on any surface, any cell, anywhere in the body where you see villi, the thing you need to automatically associate with that is absorption. Any cell that has villi on it is doing some sort of absorption. It's moving stuff from one area into another area. That's what its job is. So these little villi are going to have a core that is definitely made up of protein to help keep it in place, but we're also going to have the capillary in there along with the lacteal, because this is the site of the real absorption in the intestinal tract. So as soon as we move that nutrient out of the lumen of the intestinal tract, we need to provide that capillary or lacteal for it to go into. And so the capillaries and lacteals are going to be found in the villi of the mucosa of the small intestine. Now, we have another feature known as microvilli, which is basically our villi having villi. And these are cytoplasmic extensions of the mucosal cell. And it gives the intestinal lining a fuzzy appearance, and we call this the brush border. And what's special about this is that it contains these membrane-bound enzymes that are going to be used to finalize the digestion of carbohydrates and proteins. So carbohydrates and proteins are both really large molecules. They have lots of monomers that comprise them, and so they get broken down in stages. So you'll take some of our proteins have as many as 2,000 amino acids in it. So the first thing you have to do is unfold it, and then you start cleaving it into smaller peptide chains until you can finally get down to the very last little bit where you've got a few amino acids together, for example, and you want to cleave those into their individual subunits, the individual amino acids. And so these membrane-bound enzymes are going to do that. They're going to be the last, they're going to just finish it off. So you might have a disaccharide that's going to be cleaved into monosaccharides, and you'll have maybe a small peptide chain that'll be cleaved into amino acids, and it'll be thanks to these brush border enzymes that are doing that for us. In the duodenum, we're going to have these glands that are called Brunner's glands, and they are going to secrete a bicarbonate-rich alkaline mucus. Now, mucus is really important when we're talking about structures that are around the stomach, particularly on the receiving end of the stomach. So the duodenum is going to have to be protected. We know the stomach's protected, but the duodenum has to be protected because it's going to be receiving that very acidic chyme from the stomach. So we're going to have a lot of bicarbonate-rich secretions in this part of the small intestine in order to bring up the pH of the chyme in order to get it to a little bit closer to 7. Move it away from 2 to 4, or whatever it is, by the time it leaves the stomach, and then get it a little bit closer to 7. We're also going to have goblet cells in the small intestines, which are going to secrete mucus that adds fluid to our chyme and makes it very, watery and pasty. We'll also have Paneth cells. Paneth cells secrete lysozyme, which is an enzyme that can phagocytize bacteria. So this is part of the immune presence that we're going to see, one of the immune presences that we're going to see in the small intestines. And then we have these eye cells. And eye cells are going to secrete something called cholecystokinin. And it's going to stimulate the pancreas to release its pancreatic digestive enzymes. Now let's look at this word cholecystokinin. And there's a reason why I said it in 3 parts. This chole right here refers to the gallbladder. In fact, the term cholecysto actually refers to the gallbladder. And then kine refers to movement, as in kinesis. And then the IN ending tells us that it's a protein, which many signaling molecules are. So I can hear you now saying, well, Dr. O, why is cholecysto having to do with gallbladder? But then we say it stimulates the pancreas. the reason why is that typically when you stimulate the pancreas, you also stimulate the gallbladder. And when you stimulate the gallbladder, you also stimulate the pancreas. So cholecystokinin, yes, does stimulate the pancreas, but it also will stimulate the gallbladder to release bile. Now we know that the small intestine is going to be doing a great deal of chemical digestion, but remember it's going to be doing mechanical digestion too, because the largest amount of movement that we see in the small intestine comes by way of segmentation, which is that mashing that the muscles of the small intestine are going to do in order to take the chyme and mix it up with the digestive enzymes and further break it down. Now, after absorption, the duodenum is going to release a chemical messenger known as motilin, which is going to cause peristalsis, and that's going to be those wave-like motions that are going to help propel the residue towards the ileocecal valve. All right, and it's also going to cause the ileocecal valve to relax, and that's going to allow the residue to exit the small intestine by way of the ileum and then enter the large intestine into the cecum. Now, the small intestine is going to be the site of a lot of chemical digestion. So we start chemical digestion in the mouth. We also do mechanical digestion there. Don't do any sort of chemical or mechanical stuff in the esophagus. In the stomach, we do both. And we start the process of protein breakdown and a little bit of fat breakdown, but it all starts in earnest when we get into the small intestines. So the small intestines are about the mechanical and chemical breakdown as well as absorption. So this is really where the rubber meets the road with the thing that we think about when we think about the digestive tract. So the chemical digestion that we see occurring in the small intestine is going to be the breakdown of proteins and carbohydrates, where they've already been started elsewhere. Carbohydrates were started in the mouth, proteins were started in the stomach, and we did talk about lipase being released in the mouth and the stomach, but it's the lipid enzymes that come from the pancreas that really start the digestion of the fats. So yeah, we started in a couple of places, but real lipid digestion is thanks to the pancreatic enzymes and also bile, because remember, bile is the emulsifier. Now, when we talk about the delivery of chyme from the stomach into the duodenum, it has to happen very slowly. And this happens slowly for optimal digestion and also to give the duodenum time to neutralize that very acidic chyme. All right, because what would happen, it says here prevents water loss. What would happen is that if we were to dump in a lot of very acidic chyme into the duodenum, the pH there, the hydrogen ions there, would interact with the fluid that was in the duodenum, and it would create a whole lot of hydronium ions, and then it would start to pull fluid out of the tissues, and then it would start to pull out fluid from surrounding tissues and basically do damage in the process. So it's important for us to very carefully moderate what we allow into the duodenum and allow the duodenum time to neutralize the incoming chyme so that the tissues are not damaged. All right, we're finally in the large intestine. Now here we're going to see a thicker mucosa, also made up of simple columnar epithelium. The anal canal is where we're going to actually shift back to that stratified squamous epithelium, and it's all about the abrasion. Now, we're not going to see the circular folds that we saw in the small intestines, nor will we see the villi or any of the digestive secretions. The large intestine is not about doing any more digestion on the residue that's being dumped into it. But it does contain a bunch of deep crypts that have mucus-producing goblet cells. So we're still kind of looking at what's in there and making sure that it is fluid enough to make its way through. But I did say earlier that the large intestine acts very much like a sponge. So at some point, the large intestine is going to start really pulling out a lot of fluid and returning it back into the body, and allowing that stool to get a little bit firmer so it can be moved as a soft but formed um, lump. Lump. Let's just call it what it is, a lump. As we stated earlier, the anal sphincters, there's two of them. There's the internal sphincter, which is smooth muscle, and the external sphincter, which is skeletal. So we don't have control over the smooth muscle, but we do have control over the skeletal muscle, and both of those have to relax in order for the stool to pass. So let me tell you what you're looking at in this image. You're looking at a close-up of the anus. So up here we have one part of those rectal valves. There would actually be another one coming off here to the right, and then another one up here. It's a series of these little shelf-like structures that kind of overlap and create this sort of a labyrinth that the stool has to negotiate in order to make its way out. And that's enough to hold the stool in there and allow gases to escape, which is really what all that's about. That's what the rectal valves are for. We get into the rectum in this part, and here we can see the levator ani muscle coming off either side. So when that muscle shortens, it's actually going to lift the anus up, which will help kind of pull it open. And with the relaxation of the internal sphincter and the external sphincter, that will allow the end of the anus to open up, and the stool can make its way out through the anal canal and the anus, which is the opening. Now, the haustra, which are the little pockets, they're going to undergo a contraction about every 30 minutes. And it's the job of the haustra to start to pull water out of the residue and to ensure that we have a firm shaped stool at the end of the process. The large intestine will do peristalsis in order to move the residue along. And the large intestine does this thing called a mass movement, which is basically a big, strong contraction that happens in the middle of the transverse colon that's going to force all the residue towards that's in the remaining part of the colon, the rest of the transverse and the descending colon. It's going to force all of that towards the rectum. In addition to that, we have what's known as the gastrocolic reflex, which is initiated by the stomach and the small intestines, which will help move things along as well. So what all of this is telling us when we look at the motion of the large intestines is that when we start putting food in at one end of the alimentary canal, the other end knows that it's going to be there in a couple of hours, so it's making way for more. That's what these movements are all about. Basically, they're being triggered either by the presence or of food or by input from the stomach saying, we got more, make some room. So it's natural then that after you eat a meal, you would have a bowel movement, or if you're getting ready to eat a meal, even that could be enough stimulation that it could trigger a bowel movement. So, not unusual. Now, I've already said that in the lining of the mucosa of the large intestine, there are no glands that are going to secrete any enzymes. So the large intestine itself will not be doing any chemical digestion. However, the bacteria that live there will. So the bacteria are going to be responsible for any digestion that is occurring in the large intestines. And they're going to be doing a type of a breakdown known as fermentation. And they're going to be able to, these bacteria are going to be able to break down carbohydrates. And it's typically, it's like certain fibers and a few other things. And it's typically the action of the bacteria of the large intestine doing some sort of a digestive thing that's going to create the gas that we experience. So it's not you, it's your bacteria. So the bacteria in the large intestine is a big population. We've got about 1,000 different types of bacteria in there. And the number of bacteria in the large intestine actually outnumbers our own body cells 10 to 1. That's a lot of bacteria. And they're going to enter into the large intestine, either from the small intestines or the anus. And they're doing that in order to colonize the colon, because it's actually fertile ground for a lot of bacteria. So the bacteria are going to be doing the metabolic functions as opposed to the glands of the large intestine, because we don't have enzymes being produced there. It's all about the bacteria and the enzymes they're producing. So the bacteria are going to be doing something called fermentation, which is going to be taking indigestible carbohydrates and mucin and breaking it down. And this is what's going to be releasing the irritating acids and gases that we can find in our large intestine. Another thing that the bacteria in the large intestine are doing is they're doing some vitamin synthesis. How about that? So, for example, they will synthesize a B complex and even some vitamin K. Vitamin K is particularly important because it's a fat-soluble vitamin that we don't store in our body. We can store the other fat solubles, but we can't store this one. And we need this one in order to make clotting factors. So we make the vitamin K, it gets shipped over to the liver, and the next thing you know, we have a healthy clotting response when we break our skin open and start bleeding. The other thing that can happen with the large intestine bacterial is that they can keep pathogenic bacteria in check. So often what we see, because we have so many bacteria in the large intestine, is that the beneficial bacteria can outnumber and even suppress pathogenic bacteria. The immune system is going to be destroying any bacteria that may try to breach the mucosal barrier. So, you know, we've got some immune function just kind of by a numbers game based upon healthy bacteria, and also some, lymphoid tissue that is going to be found in the submucosal layer of the large intestine. Now, as the years have gone by, more studies have been done on the gut microbiome and overall health. And there is mounting evidence that supports that healthy gut bacteria mean a healthy human being. And it can affect everything from body weight to susceptibility to various diseases such as diabetes, fatty liver, and even atherosclerosis. It can even have an effect on your mood and cognition. So having a healthy gut is super important to being a healthy human being. And functional medicine is starting to look at that and utilize healthy gut bacteria, or utilize healthy gut and looking at healthy gut analysis in order to help move a very sick patient towards health. And I suspect it's going to be rather routine, maybe in the next couple of decades. Kind of a nifty approach when you think about it, just using the body's own defenses to restore health. So let's talk a little bit about the accessory organs of digestion, and we're going to start off by talking about the liver and how it's put together. Now, the liver is a highly organized organ, and the functional units are going to be these liver lobules, which are hexagonal shaped structures. And that structure is really important because at the corner of each hexagonal lobule is going to be this portal triad. So there's some vessels there at the corner of each of these hexagonal little lobes. The cells of the liver are called hepatocytes, and they are going to filter and process that nutrient-rich blood that's being fed to it by way of the hepatic portal vein. Because remember that we're going to take the blood that's coming from the intestines after it's been had all the nutrients dropped into it, and we're going to shuttle that directly over to the liver before we return that blood to general circulation. The portal triad that we're going to find at the corner of each of these lobules is going to contain a branch of the hepatic artery, which is bringing oxygen-rich and also nutrient-rich blood to the liver. the hepatic portal vein, which is bringing oxygen poor, but also super nutrient dense blood to the liver, and then also a branch of the bile duct. Now, the blood that's coming in, either whether it's from the hepatic artery or the hepatic portal vein, is going to be filtering into the lobule, and it'll eventually make its way into what's called a central vein, which will drain the lobule. Now, as the nutrients are being pulled out and the fat's being pulled out, the hepatocytes are going to be making bile, and that's actually going to drain towards the corner and be collected by that branch of the bile duct. So there's these little bile canaliculi. We've seen that word once before over in A&P2, which are basically these little channels, little canals, that are going to collect the bile that as it's being made by the hepatocytes, and they're going to usher it in the opposite direction of blood flow and get it out to that corner, and eventually drain it into either the right or the left hepatic bile duct, and then it'll eventually make its way out of the liver. Now, the capillaries that are feeding into the liver are, of course, going to be very weepy. In fact, the capillaries coming in are known as the sinusoidal capillaries. So they're the weepiest and leakiest of our capillaries. So this is what we're going to see here in the liver. So the blood that's coming from the hepatic portal vein and the hepatic artery are going to percolate from the corner of the hexagonal portal triad, and they're going to basically make their way through these sinusoidal capillary beds and then ultimately empty into the central vein. Now, whenever we have these sinusoidal capillaries in place, we're always going to have an immune response present, because with these big holes in our capillaries, that's an avenue for pathogens to gain entry. So in the liver, we have these macrophages that live there. They're hepatic macrophages. They're also called stellate because they look like stars with all the rays coming off a central region. And they're going to live in the liver sinusoids and remove debris and old red blood cells and other things that don't necessarily need to be returned to the blood in the form that they show up in the liver. So here we have an example of a liver lobule, and we've got a portal triad here, and then one here at this corner, this corner, this corner, and of course at this corner. So we have blood coming in from the hepatic artery, and then blood coming in from the hepatic portal vein. And then as we see, it's going to branch, and that blood is going to percolate through the capillary, those sinusoidal capillaries, so that the nutrients and debris and whatever else needs to be cleansed by the hepatocytes can have all of that process happening. And then the blood, all of it, whether it's coming from the artery or the vein, are going to drain into this central vein that it runs through the middle of each lobule. All right. Now, meanwhile, these hepatocytes are going to be making bile. And there's going to be all of these little bile canaliculi that are going to collect it, and they're going to drain it into these little bile ducts right here, and then these eventually are going to merge into either the right hepatic duct or the left hepatic duct and drain the bile away from the lobule. Because of the type of activities that the liver cells are doing, the hepatocytes are doing, if we were to look at one of those, the thing that we would see is that they have tons of rough ER because they're doing a lot of protein synthesis, and they're going to have tons of smooth ER because they're doing a lot of detoxification and fat metabolism and carb metabolism as well. And because we have tons of this ER, we're also going to have a fairly elaborate Golgi apparatus to help package and ship all of the products that are being made by the different endoplasmic reticulums. Because we're doing filtration in the hepatocytes, we're going to have peroxisomes, because remember, those are organelles that are associated with the smooth endoplasmic reticulum and detoxifying some of the metabolites that get created over in that part of the cell. And then whenever we're moving things around with the Golgi apparatus and ushering things out of the cell, By way of exocytosis, we have to have a lot of ATP to do that. So we're going to also have a lot of mitochondria. Now, hepatocytes are super busy. They can produce up to 1 liter of bile a day. That's a lot. They can process any number of bloodborne nutrients. So, for example, they can take glucose and store it as glycogen. They're responsible for making almost all of the plasma proteins except for gamma globulins, which are made by the B lymphocytes. And they are also dealing with fats. So they can take fats and manipulate them and turn them into phospholipids and do all kinds of crazy things with fats. They are the liver will, or the hepatocytes will store fat-soluble vitamins, and the hepatocytes will also perform detoxification. So they will take metabolites from cellular metabolism and then convert it into something that is less lethal in the body. So at any given moment, the liver is doing upwards of 500 different metabolic activities. It's a very, very busy organ and a very important organ. And amazingly, an organ that can regenerate itself. Crazy. Now, we talked a bit about bile when we were in the blood unit, so let's go ahead and talk about it here again. We know that it's made in part from bilirubin, which is that pigment that comes from the breakdown of red blood cells, particularly the heme in the hemoglobin, and bile salts, which are largely cholesterol-based molecules. And that this cholesterol is going to help digest fat, because in the world of chemistry, like dissolves like. The other thing that we're going to find in bile is triglycerides, phospholipids, and electrolytes. So kind of some nasty stuff, but it's important and it has a lot of useful things in there. And I'm glad I have it. Now, the story of bile is actually a great story about recycling, because we make bile from recycled molecules and basically parts of red blood cells, and we actually recycle large parts of bile from the small intestine. So the story of bile is told by way of the enterohepatic circulation. So this basically is the story that bile salts will enter into the duodenum and do fat emulsification, and then later be reabsorbed by the ileum, the last part of the small intestine, and then returned to the liver by way of the hepatic portal vein. And then they'll make their way back through the bile canaliculi and through the duct network, and then into maybe the gallbladder, maybe not, and then re-enter the small intestine again, only to be pulled back out at the right time. And this is a really efficient system. About 95% of the bile salts get taken back up and recycled. So at any given time, we only need to create about 5% to replace what gets lost in the intestinal tract. So it's a kind of an interesting system. But yeah, recycling. Now, I know we talked about the pancreas when we were in the endocrine system, but it's also an organ that does exocrine function as well. And its exocrine function shows up here in the digestive system. Now, the pancreas is mostly retroperitoneal, which means it's going to be outside of the peritoneal cavity. And we're going to find that it has this rounded end called the head that is tucked up next to the duodenum, and then it's got this little pointy tail that's going to be at the opposite end, and that's going to be towards the spleen. The exocrine function of the pancreas, making the pancreatic juice, is, I'm going to say, necessary for life, because it is with these digestive juices that we're capable of digesting all different sorts of macromolecules. And without this, we just wouldn't be able to do that job very well at all. Now, there are these cells called the Acini cells, which are responsible for making these enzymes. They produce what are known as zymogen granules, and you'll notice that ogen ending there. That's because these enzymes are released in an almost ready to go state, because we don't want these enzymes released ready to go because they would digest the pancreas. So these are going to be in an almost ready to go state, and once they get into the duodenum, there will be other substances there that will activate these almost ready to go enzymes. So we could call these proenzymes because they're almost ready to go. In the pancreas, there is an extensive duct network that will collect the zymogen granules from the acini cells. They're called acinar cells, and collect everything and eventually deliver the pancreatic juices to the duodenum. Some of the duct cells are going to produce water and bicarbonate because we need to eliminate that strong acid that's coming in from the stomach. And we know that the pancreas does an endocrine function, and that's going to involve insulin and glucagon and the management of blood sugars. So I've said this a number of times already, but the pancreas makes the most potent digestive juices of the entire digestive system. Even though it's an accessory organ, it is one that is supremely important. What are we going to find in pancreatic juices? Well, we're making up to 1.5 liters of it a day, and it's going to contain water and an alkaline solution, mostly bicarbonate, in order to neutralize the acidic chyme. It's going to contain electrolytes. There's our electrolyte bicarbonate right there, and then digestive enzymes. So proteases, which are going to break down proteins. Amylase, hey, we had some of that in our saliva, but not a lot. This is where we're getting the big load right here. That's going to digest carbohydrates, lipases for lipids, and nucleases for nucleic acids. So when I say that the pancreatic juice is the most potent, it's because it can digest all 4 of these macromolecules that we're going to find in food. Now, as I said, these enzymes are going to be largely in an inactive form and are going to have to be activated once they reach the duodenum. So in the duodenum, we have enteropepsidase, which is an enzyme bound to the plasma membrane of the duodenum, and it's going to activate the pancreatic protease trypsinogen and convert it into trypsin. So basically we have these membrane-bound enzymes in the duodenum that once the pancreatic juice makes its way in there, those membrane-bound enzymes will start to activate the juices. And of the juices that get activated, the enzymes that get activated is trypsin. And trypsin is kind of an important one in the digestion of proteins. Now, once trypsin is activated, then it can activate other things. So, for example, it can activate more trypsinogen. So, if there's a local trypsin that's been newly activated and there's any trypsinogen around it, it will activate the trypsinogen. So that local trypsinogen doesn't have to make its way over to the mucosal membrane to make contact with an enzyme over there. It can just be activated by trypsin. We have a couple of other types of enzymes here that start off in their proenzyme state and then become something else. We have carboxypepsidase as well as chymotrypsin. Now I know we haven't talked about what either one of those do, but we can probably guess just based upon it being activated by trypsin that it's probably going to be breaking down either protein or carbohydrates, because that's what's in the digestive juices. Now, this is a very basic picture of the pancreas, but I want to go over this very briefly. So this is the tail of the pancreas, and this rounded region is the head, and it's going to be nuzzled into this curvature of the duodenum. And what you see here in green is the extensive duct network that's going to be collecting all the digestive enzymes that are being made by the acinar cells. And it's going to be collected and contained up here in an expanded region known as an ampulla. And then in here in the duodenal wall is going to be a sphincter that will open and close as the pancreatic juices are needed. So that's a little bit about the pancreas. So there's going to be a main duct that drains everything. There's a little holding area known as an ampulla, and then there on the other side of that, where it embeds into the wall of the duodenum, is going to be a sphincter that will open and close and allow those pancreatic juices in as they are needed. Now, earlier we talked about cholecystokinin, and I stated that even though it has the word cholecystic in it, which makes you think about gallbladder more than pancreas, it's going to stimulate both. And there's a reason for that. And it's because there comes a point where the bile duct coming from the gallbladder and the liver is going to join up with the duct coming from the pancreas. And so that when we release the pancreatic digestive juices into the duodenum, we're also going to be releasing bile. So typically we're going to see both of those glands stimulated at the same time, the pancreas and the gallbladder. All right. So where the bile duct and the pancreatic duct unite in the wall of the duodenum, that's going to form this enlargement I called an ampulla. Now, I've seen it called the hepatopancreatic ampulla, but I learned it as the ampulla of Vater, named after the German anatomist who discovered it. And the sphincter that is going to open and close and allow the fluids into the duodenum is can be called the hepatopancreatic sphincter, or as I learned it, the sphincter of Oddi, named after the Italian anatomist who discovered it. I like those names. Now, because the movement of the pancreatic digestive juices into the duodenum is super important, there's actually an accessory pancreatic duct that will bypass this ampulla, the ampulla of Vater, and attach directly to the duodenal wall. So if there were to be an issue with the ampulla of Vater or maybe the sphincter of Oddi, the digestive juices could circumvent that and still get into the duodenum, and we would survive. So regulation of bile and pancreatic secretions are going to be controlled by very similar signaling molecules, and we've already talked about cholecystokinin, and then there's another one known as secretin. So these are a couple of hormones that are going to cause the bile to be released by the gallbladder and the digestive enzymes by the pancreas. So the thing that we know about bile and the pancreatic secretions is that they're going to, their job is to break down fat and proteins and carbs and nucleic acids. That's what we know about them. So when are we going to release these secretions? Well, we can, the release of these secretions can be triggered if there is a large amount of bile salts that get returned. So that could be a trigger for the release. So if we think about it, we've just maybe digested a bunch of food or finished digesting a bunch of food, and maybe there's more food on the way. The body might be interpreting the return of those bile salts as another imminent feeding time is forthcoming. We have intestinal cells that are going to release that secretin that we know will stimulate the release of bile and pancreatic juice. And they get triggered to release that secretin when they are exposed to hydrochloric acid and fatty chyme. So when the cells of the intestine are looking at that, they're like, whoa, we just got something from the stomach, and man, look at all of that fat. We better get something in here to work on it. And so, of course, they're going to call for help from the bile and from the digestive enzymes. Now, under most circumstances, the sphincter of Oddi, the hepatopancreatic sphincter, is going to be closed, which means that the bile and the pancreatic enzymes are going to be on the other side, hanging out in the ampulla of Vater. And the only time that the sphincter of Oddi is going to be triggered to be open is if there is active digestion happening. So all of this secreting of bile and digestive juices has to be coordinated around all these triggers that are associated with the intake of food and the initial digestion of food. Now here's another image that includes not only the pancreas but also the gallbladder, because I wanted to show you these structures here. So we know that this is our pancreas here. There's our tail, there's our head, here's our main duct draining into the ampulla of Vater right here. And if we come up here to our gallbladder, there's our cystic duct and then our bile duct coming in. And oh look, it too has an attachment to the ampulla of Vater. So when I said that these two substances are going to mix and then be released, I meant that they're going to be mixed and released at the same time, right here through the sphincter of Oddi, all right, into the duodenum. And you'll notice that here's our main pancreatic duct, and for the most part, we have this big connection here with the ampulla of Vater, but you'll notice that we got this little detour right here going directly into the duodenum. So that's the accessory pancreatic duct. So that if something were to happen here, I don't know, maybe a little stone made its way, boop, got stuck in there, we can still get digestive enzymes here. Maybe not so much bile, but definitely digestive enzymes from the pancreas. Now we've already defined digestion and absorption, and what we know is that digestion involves the breakdown of the food that we've ingested, and we're turning it into those molecules, whether it's a glucose molecule or an amino acid or maybe a building block of nucleic acid. And it's only these molecules that are small enough to be transported or absorbed across the wall of the intestine. So we can't move big sugars out. We can't move big peptide or even small peptide chains out. We have to break these food substances into their constituent parts, their monomer parts, if you will, in order to get them across the intestinal wall. And the other thing is that, you know, our body knows what it needs, and it's going to take these building blocks and it's going to reassemble them in a way that's beneficial to us. So digestion is basically taking the big thing, breaking it down into the small thing, and making the small thing available to be moved across the membrane in an act called absorption, so that the cells can take it and do what we need with it. Now we have enzymatic hydrolysis or hydrolysis. We know that digestion is largely a catabolic process that breaks down big molecules into small molecules small enough to be absorbed across that mucosal membrane. And that there's going to be intrinsic and accessory glands that are producing these enzymes that are involved in all of this. This is what we talked about. We talked about the brush border with those enzymes embedded. We talked about the enzyme activators in the duodenum. We talked about the enzymes that are made by the pancreas. We talked about fat emulsion being made by the bile. That's what we're talking about when we say the intrinsic and accessory gland enzymes being involved with digestion. Now, the enzymes are doing this through an active hydrolysis, and you might remember hydrolysis or hydrolysis from AMP1. In order to break down a large molecule, you have to create an exposed area when you tear that molecule apart. And we don't want to leave an atom at the end of a molecule with nothing attached there. That creates an imbalance. So what Mother Nature does is she'll take a water molecule and she'll rip off a hydrogen and leave the hydroxide as it is, and she'll use those to put in that place where the molecule, our big molecule, was torn apart. So that's why it is a hydrolysis or a hydrolysis reaction. We cleave a water molecule in order to patch the ends of the big molecule, the macromolecule that we're tearing into parts. You might remember the business about an enzymatic reaction. We have a degradation reaction, which is what this is called, where we've got our big molecule known as a substrate and our enzyme. And here we're taking our big molecule and we're breaking it down into smaller molecules. So this is a degradation reaction. This is a catabolic reaction. So what we mean by a hydrolysis reaction is that right in this area where we separate these two, we would put a hydrogen right there and a hydroxide right there, something like that. That's what we're talking about, this being a hydrolysis reaction. So there's our water molecule. We're going to cleave it in two, and we're going to put a hydrogen, that's our hydrolysis, and we're going to put a hydrogen there and a hydroxide right there. All right, now everybody's happy and balanced. So this is why the catabolic reaction, the degradation reactions, are classified as hydrolysis reaction. Hopefully that rings a bell. The mechanisms of transport of substances, moving these things from the lumen of the intestine into the bloodstream. And when we look at what's happening in the intestines, we have tight junctions that are ensuring that molecules have to pass through the epithelial cell rather than between them. So we want our intestinal lumen, we want the mucosal lining to be, regulatory, and that we control what can come out of the intestinal lumen and into the blood supply. So that's what those tight junctions are telling us. Tight junctions are the special contact points that mean that substances cannot slip in between cells, that they actually have to be moved through the mucosal cell, epithelial cells, in order to get into the blood supply. And those movement methods are going to be things like passive absorption, channels, pumps, or even co-transport of some sort. So all of those different mechanisms that we talked about earlier. Now, lipid molecules can be absorbed passively through the membrane, but other polar molecules have to be absorbed by an active transport mechanism. So we typically going to have a little bit of ATP expended in order to make a large number of things move across the epithelial cells and into the blood supply. When it comes to carbohydrate digestion, only the simplest sugars can be absorbed across the mucosal barrier and enter into the blood supply. Our big carbohydrates, things like starches or even disaccharides, which are two sugar rings bound together, these get broken down into oligosaccharides and disaccharides, or smaller carbohydrates. And this whole process starts in the mouth with that amylase that's found in saliva. Now, eventually, all of these large carbohydrate molecules, the starches and such, are going to be further broken down into lactose, maltose, and sucrose. And then finally, those are going to be broken down into the monosaccharides. So this is where we're going to get glucose. Or we could also end up with fructose and galactose. Now, the interesting thing about this is that our body only wants to use glucose. So why are we bothering to break down fructose and galactose? Because the liver can take those other two sugars and turn them into glucose. So that's a good reason why we're going to take this blood that's coming from the intestines with all of this other stuff in here, like the fructose and the galactose, and shuttle it over to the liver so the liver can do a few things that need to be done in order to make the nutrients that are in there usable by the tissues. So let's say that we had a big long glucose chain. Amylase is going to come along and it's going to break this big chain up into a few smaller chains. And then amylase will come along again and break down those additional unions until we end up with our single sugars, our monosaccharides. So this is what's happening when we're doing digestion of carbohydrates. We can get proteins from a number of places, not just necessarily from our diet, because keep in mind, enzymes are made of proteins, and many of our cells are going to contain proteins. So we could be getting proteins from other parts of the body through a natural breakdown process. But wherever they come from, our proteins are going to be broken down first into large polypeptides, and then into small polypeptides, and then finally into the amino acids, which are the building blocks of proteins. Digestion of proteins begins in the stomach when pepsinogen gets converted to pepsin due to that very low pH. Now, we release a little bit of lipase in the mouth and in the stomach, but we don't start the digestion of lipids in earnest until we're in the small intestines and we have that pancreatic digestive lipase to work on our fats. So the first thing that we have to do with our fats is we have to do emulsification. So we have to take triglycerides and their breakdown products and get these into a smaller globule basically is what is happening here. Because remember, we're largely in a water environment, and these fat globules are going to want to stick together. They're going to be basically repelled by water molecules, and they're going to be forced together into these large globules. So we have to be able to break them down, first of all. And then along comes our pancreatic lipase and starts breaking them down into monoglycerides, plus a couple of free fatty acids. So we went from, triglycerides down to a monoglyceride. One of the other things that can happen is we end up creating this structure known as a micelle. And what happens here is that we are basically taking products from digestion and coating them with bile salts and lecithin so that they can be easily dropped into the blood supply and transported around the body, because that's basically what a micelle is. It's an escort structure for fats. We can it plays nicely in water, but we can tuck the fats inside and transport them around. Now, lipid products can leave the micelles and cross the epithelial membrane by way of diffusion, because remember, membranes are largely made of fat, and fat-based products are going to be able to slip past them quite easily. So just to review what fats look like without having to go into the entire molecular structure, you have this glycerol backbone right here, which is a carbon, hydrogen, oxygen molecule, and attached to it are three fatty acids, which is a carbon-hydrogen chain. And when we take a triglyceride and turn it into a monoglyceride, we lop off a couple of these fatty acids, leave one intact, and then attach a phosphate to it and turn it into a monoglyceride. And with this scenario, we can take it and we can create this structure. And you'll notice that the phosphate head is pointing out and the fatty acid tail is pointing in. So the phosphate head will allow this structure to integrate nicely in a water environment, and we can tuck fats inside and transport them around. So that's the beauty of a micelle. Now, another structure that we can make out of fats is called a chylomicron. And this is a larger structure comprised of lipids, and we can basically tuck even more fats inside of this thing. It tends to be shored up with some lipoproteins, and because it is a larger structure, so it needs a little bit more integrity given to its membrane. But chylomicrons are going to be transport structures, and they get exocytosed from the bottom part of our simple columnar epithelial cells that are making up our mucosa, and these structures will enter into the lacteal and be transported around through the lymph and then eventually be dropped into the blood. So here we see a chylomicron, and you'll notice that we have the phosphate heads and the fatty acid tails. Phosphate heads are pointing out, which is what allows us to integrate nicely in water. And we also have some apolipoproteins studded in here to give this a little bit of integrity. And then packed in here are a bunch of triglycerides. So this is the structure that we talked about being too big to enter into the blood supply, but being able to be taken up by a lacteal and making its way through the lymph and then ultimately being returned into the venous blood supply. This is that structure right here. So let's take a look at some homeostatic imbalances, and we're going to start off by talking about GERD, gastroesophageal reflux disease. Now, when the stomach contents routinely enter into the esophagus, this is when we get this problem. And what the patient is going to experience is a burning sensation or maybe a pain in the chest. There's going to be a lot of regurgitation, a lot of burping, sensation like stuff is coming back up, maybe a lump in the throat. They might even have irritation of the larynx due to the gastric contents making its way all the way up to the voice box and creating irritation. This person is going to be uncomfortable when they sleep, because as soon as they lay down, they're going to have the contents of the stomach washing into the esophagus. Now, why does this happen? Well, it happens because that lower esophageal sphincter might be weakened or damaged. So what puts a patient at risk of getting GERD? Well, obesity, Having a hiatal hernia, which is that condition where part of the stomach pokes back through that opening that the esophagus penetrates in order to attach to the stomach. That can create a problem. Pregnancy, because pregnancy is going to move everything around to accommodate that baby. If you have a connective tissue disease and you can't keep your organs where they're supposed to be, that can also create this scenario by allowing the stomach to move around and kind of like a hiatal hernia. Smoking, eating large meals, eating before bedtime and then laying down, coffee, alcohol, medication. So there's a number of things that can lead you down the pathway of GERD. So how do you treat it? Well, you can take medication for it, and you can try to reduce the stomach acid. You can also do simple things like elevate your bed so that you're not sleeping in a completely horizontal position. So elevate your head so that your head's up a little bit so the stomach contents don't wash up so far up into the esophagus, things like that. Change your diet to deal with that acid, and that might mean, unfortunately, cutting out coffee or alcohol, or eating smaller meals, not eating before bedtime. There's a number of things that you can do to head this off and reduce the chances of it. Now, celiac disease is when the patient experience an immune response by eating gluten, and it's going to cause a lot of tissue damage in the small intestines. This is an inherited condition, and the genetic code has to line up just right for the person to get it. But interestingly enough, we're starting to see this more and more in the population. When I was in school 20 years ago plus, we were taught that we would never see a patient with this disease in our practice. And by the time I was in practice, I was seeing all kinds of people with it. So I don't know if it was underreported or if now we have a greater incidence or there's something in the environment triggering its emergence. I don't know how to explain that. But yeah, it's kind of funny that I was taught initially that we would never see it, and then by the time I graduated 4 years later, it's everywhere. So what does it look like? Well, it can look like a number of things, but the most common set of symptomology is going to include diarrhea and constipation, swinging back and forth between those two, fatigue, weight loss, and in some cases weight gain. Because keep in mind, we've got a disruption of the microbiome in this disease process, and when we have that disruption of the gut bacteria, that can make it really difficult to lose weight, or can make it very difficult to absorb nutrients, and the person can waste away. Other symptoms that the patient may exhibit are anemia, osteoporosis, skin rashes, headaches, even some cognitive issues like depression, cloudy thinking. There's a number of things that can be associated with this sort of allergy and gut disruption. And the treatment is, first of all, to avoid gluten, get it out of your diet. And it's not on this slide, but I'll tell you, the second part of treatment is to start feeding your body what it needs to recover. That's an important step that often gets left out in the treatment protocol, because sometimes if you've been eating the gluten for too long, you've sustained a lot of damage, and simply cutting out, simply eliminating is not enough. You then have to be very mindful to feed your gut and your body the things that it needs to rebuild. Irritable bowel syndrome is a condition of the large intestines, and it also has bouts of diarrhea and constipation, can swing between the two, typically abdominal pain with a great deal of bloating. The cause of IBS is largely unknown, but it's believed that stress is a contributing factor, as is maybe some sort of bacterial overgrowth or an overreactive nervous system, which could be due to stress. How do we treat this? High fiber diet, adequate fluid intake, exercise, and sleep, making sure you've got good sleep. Medications might be used to relax the large intestine and manage stress, or reduce bacteria if for some reason there's some sort of an overgrowth. Ulcerative colitis is an inflammation and ulceration of the large intestine and the rectum. This patient will have diarrhea, often with blood or pus in the stool. or the fluid. There's going to be a lot of abdominal and rectal pain because of the cramping and the ulceration. This patient often has defecation urgency and may lack the inability or may lack the ability to actually hold the stool because of that urgency. This patient often has weight loss and fatigue as well as a fever. The cause of ulcerative colitis is largely unknown, but it's believed to have an autoimmune component to it, or maybe even some hereditary predispositions. The treatment of this involves medication to manage the symptoms, and surgery may be in order if there are particularly bad areas of infection. There's a number of ways that we can get to liver disease. You might have hereditary factors that are not in your favor, but are in favor of liver disease. Lifestyle certainly plays a role into this. There may be some pathology, underlying pathology, that is contributing to liver disease, or maybe even a pathogen. So when you have a liver disease, one of the things that you're likely to experience is jaundice. So keep in mind the liver is going to be taking that bilirubin out of circulation in order to put it into bile. But if the hepatocytes are damaged, then the bilirubin stays in circulation, and that's what creates the jaundice. The other thing that may be happening is abdominal pain, swelling of the ankles and legs, itchy skin that bruises easily, pale stools, and dark urine. And all of these are largely due to the liver not being able to filter the blood like it needs to. And so we end up with a backup of fluids. We end up with maybe recirculating of toxins and debris that the liver would have already removed from the blood, which can lead to the itching. And the pale stool is also problematic of a liver that's not functioning, as is the dark urine. So there's a couple of hereditary factors that can lead to liver disease. One of them is hemochromatosis, which is when you have an excessive iron absorption. And you will encounter patients who have this issue, and it's important that they get good supplementation, but they're going to be looking for those vitamins that don't have iron in it. So if your office is going to be providing any sort of nutritional support, you have to be mindful that there's going to be a portion of the population that is going to be wanting to take the vitamins without the added iron. There's also Wilson's disease, which is an excessive copper absorption issue. So it's interesting that we have two disease processes based upon hereditary factors, and both of them have to do with an excessive amount of metal uptake in the body. Cirrhosis is a liver disease that involves scarring and damage to the liver, and it can be caused by a number of diseases that affect the liver. Commonly, hepatitis is one that can lead to cirrhosis, as is excessive alcoholism. The symptoms of cirrhosis include those seen in liver disease, plus spider veins in the skin and a redness in the palms. So those are two trademarks of cirrhosis. Now, treatment of cirrhosis is going to depend upon the cause and the degree of damage to the liver. And management of cirrhosis can be complicated because this is a very challenging progression to deal with once it gets started. Hepatitis is inflammation of the liver caused by 5 different viruses. And when someone has hepatitis, you're going to be looking at someone who has jaundice. They have the dark urine and the pale stools, fever, fatigue, nausea, achy joints. So all the things that we would expect to see in someone who has liver disease. And the treatment of hepatitis is going to depend upon which virus is involved. If you have hepatitis A, there is no treatment for that. If you have hepatitis B, it can be successfully treated with drugs. Hepatitis C can also be treated with medication. Hepatitis D and E include using drugs with interferons, remember those from the last unit, as well as drugs used to treat type B hepatitis. So those are our 5. Lactose intolerance is when you have the inability to digest milk sugar. And the reason for this is that you don't have the necessary digestive enzyme known as lactase in order to break that milk sugar down. Symptoms of lactose intolerance are going to be abdominal pain, bloating, gas, nausea, and diarrhea. And lactose intolerance is kind of funny. It can occur naturally because over time it is possible that your brush border isn't making lactase as abundantly as it once did. So you can actually develop a little bit of an intolerance over time. Maybe there's some sort of a damage that was done to the small intestine, such as what you see with celiac disease, and maybe those enzyme producing cells were damaged and now you can't make the lactase. Maybe your genetics didn't ever allow you to make enough lactase for it to be meaningful for you to digest lactose. So there's a number of things that can lead you down the pathway of lactose intolerance. So what do you do for this? Well, you can limit your dairy products. Although I have to admit, I read an article the other day that said that a small amount of dairy, even in the lactose intolerant patient, is actually good for the gut flora. So just a little bit. I mean, it was very little, not enough to create all the symptoms of lactose. So you wouldn't want to be drinking like an entire glass of milk. It might be, something as small as, a quarter cup of milk, something like that. You can try the lactose-free alternatives if you have to do dairy, or you can take the enzymes. And then generally you just want a healthy diet because you don't want to be aggravating that intestinal lining any more than you have to. Constipation is when you have infrequent bowel movements that can last up to weeks. Symptoms here can include fewer than 3 stools per week. The stools tend to be hard. The patient feels like there's a blockage or a need to still empty the bowel. And the causes of constipation can include an obstruction. Maybe there's cancer involved. There could be a neurological issue such as a stroke or a spinal cord injury, and those neurological signals to actually move things along just aren't getting to the large intestine and the rectum. Maybe there's weakened or spasmed pelvic muscles. Diabetes can lead to constipation, as can hypothyroidism. So the treatment of constipation is going to depend on the cause, but it can include things like a high fiber diet, exercise, because moving around is always good for stools that aren't willing to move very easily, stool softeners, drugs, surgery, enemas. So there's a number of things that can be done to try to help move the stool along in a case of constipation. Diarrhea is the other end of the spectrum, where the stools are not hard and immovable, but loose and very watery and willing to come out very easily. The symptoms of diarrhea include abdominal pain and bloating, nausea. You might even have blood or mucus in the stool if the lining of the large intestine is particularly irritated. There's going to be a sense of urgency around eliminating that watery stool. There could be fever. There could even be vomiting. Now, the causes of diarrhea, there's a lot of different causes. You could have an infection such as a flu. You might have a food intolerance that causes irritation leading to diarrhea. Medications can lead to diarrhea, and any number of digestive orders can also lead to diarrhea. Now, the treatment of the diarrhea is going to depend upon the cause, but treatments can include antibiotics, dietary changes, medication adjustments, if that's what's creating the problem, and managing any other disorder that might be contributing. So if you have a flu and you've got the diarrhea that accompanies that, then you just let that run its course, make sure you're hydrated, and then after a few days you should be done with it. If you've got a food allergy, eliminate the food and give your gut time to heal before you do any sort of dietary challenges. So that's what we mean by, you have to treat it according to the cause. And with that, we are done with Unit 7. Thanks for watching.